Hi, my name is Enrico Barrozo. I went to Augsburg College in Minneapolis where I earned a Bachelor's of Science degree in biology. I've always been interested in the human condition. My mom passed away in April of this year, and she was chronically ill for nearly 10 years. I was going in and out of hospitals and always being around physicians. I was constantly asking questions, trying to find out what was wrong with my mom. This drove me to become a biology major. At Augsburg College, I worked with my undergraduate mentor, Dr. Matthew Beckman, where I learned that I have a passion for research. I'm driven by the possibility of discovery and innovation in my daily life as a scientist, and I've learned that I find research rewarding and intellectually stimulating. Before this year, I would have said I'm interested in genetics. However, genetics is a broad field, and it's much more of an approach. I'm interested in personalized medicine, and in the future, I want to use pharmacogenetics, which is using a person's unique genetic code to design therapies and prevent disease. My long-term goals are to get a PhD where I will learn basic science and techniques that will be essential for my future. Next, I'll get postdoctoral training, and that will determine what I'll do for the rest of my life. Eventually, I want to be doing cutting-edge research in my own lab, researching medical development or pharmaceuticals. While at Augsburg College, I was passionate about a lot of things. I had to manage my time effectively. I was a captain of the football team and doing research when I wasn't in classes. I was also working 2 to 3 part-time jobs and working towards getting my degree. Last spring, I was awarded with the Student Leader of the Year award for my accomplishments at Augsburg. This meant a lot to me, but after graduation, I wanted to redefine myself and focus more on my research. Here, in prep at UGA, I'm living the life of a graduate student. I work 40 hours in a lab, take graduate-level courses, and I'm learning essential techniques that will help me hit the ground running when I'm accepted into a PhD program. This post-baccalaureate training in infectious disease research will provide evidence that I can be a successful graduate student. I've shown that I'm fully dedicated to research and to becoming a scientist. My faculty mentor is Dr. Biao He, and I work with Shannon Fan, a 4th-year graduate student. I'm studying respiratory syncytial virus. RSV infects everyone by the age of 2, and there's currently no licensed vaccine. Symptoms result from lower respiratory infection, and virulence of RSV increases when infecting infants, the elderly, and the immunosuppressed. Specifically, my project is investigating the interaction between RSV phosphoprotein P and host kinases on their RSV life cycle. The RSV-P protein is a heavily phosphorylated protein that stabilizes the RSV polymerase complex. It is hypothesized that P phosphorylation by host kinases is important for viral transcription and replication, but the precise role is unclear. Our goal is to determine which serine and threonine residues in RSV-P are crucial for RSV replication. Our approach includes designing primers to create plasmids encoding RSV-P with serine and threonine residues mutated to alanine. This will prevent phosphorylation of those sites in the P protein. We will use standard cloning techniques such as polymerase chain reaction, or PCR, splicing by overlap, digestion, ligation, and transformation into an E. coli plasmid. Next, we will be sequencing the plasmids with the P protein inserted to make sure the correct mutations have been introduced. Then, we will be using these plasmids to perform a luciferase reporter-based mini-genome assay to measure activity of the RSV replication complex. This assay will help us determine if the mutations have an effect on the transcription and replication of RSV. My name is Enrico Barrozo, and I am a Prep at UGA scholar.